Okay, well, it's a pleasure to have this opportunity to share some of our work with you. Koei gave you a brief introduction. We'll try to fill in some of the details there. So this is really a research in progress talk. We are going to tell you about a study that's been ongoing for about the last 5 years. And we're in an analysis phase now, and we're looking forward to hearing what you think about what we've found so far, and to get any suggestions that you might have. There will be a couple of natural points during the talk where we'll pause to open the floor for Q&A. So you need not hold questions to the end. If you have any, we'll sort of pause and see if anybody would like to ask a question at those defined points. So. Okay, there we go. Okay, so this project has been a lot of fun for us. It's been a highly interdisciplinary project, bringing people together from Duke University, from Northwestern, and from the University of Massachusetts Medical School. You know, we're only going to be able to highlight a few of the people's work in the talk today, but Sarah Armstrong and Pat Seed, bring clinical expertise to the project. Chris Newgard, Beth Shank, and Spotty Shaw have brought their metabolomics and metabolic disease expertise. Folks like Lawrence David and Holly Dressman and Josh Granick have brought expertise in computational aspects of microbiome analysis. Rafael Valdivia and Ling-Chong Yu at Duke have brought expertise in bacterial genetics, and then folks like myself and Jiling Shen have brought some expertise in notabiotic animal models. And all these tools have been really useful in the work that we're going to describe to you today. And so these are the faculty that are involved in the project, but the real credit goes to the various people in our teams that have really been carrying this work out. This is a near complete list of all of the folks that have been involved in either recruitment of patients, or sample handling, or data generation, or data analysis. And we're going to be hearing from our fearless leader here, Jessica McCann, who's really led a lot of the science throughout this project and helped coordinate a lot of the activities across this very large but highly cooperative group. So so this project, which we call the Pediatric Obesity Microbiome and Metabolome Study, or POMMS, had several guiding principles and study objectives. So as many of us are probably aware, adults with obesity do have metabolic and clinical signatures that are distinct from healthy weight controls, but are also distinct from adolescents that also have obesity. And so there are a lot of questions then about how obesity may differ between different life stages. There also exists a really interesting body of work showing that mostly coming from obese adult donors, that stool from those donors can at least temporarily transfer obesity phenotypes to germ-free mice, which I think really got a lot of people interested in the mechanistic roles by which gut microbes might contribute to obesity and associated metabolic processes. But a lot of those mechanisms still remain unresolved, but the causal contributions of the adult gut microbiome to adult obesity seems to have a strong premise, but again, we don't know if that also holds true in adolescents. And again, in general, we know a lot less about microbiome and metabolic signatures of obesity during the adolescent period. And so what we're really hoping with this project is that by studying adolescents with obesity, we can identify some metabolic and microbial signatures and targets that might be useful in new therapeutics and help increase healthy life years in our patients. So we have a website up. This is the address here. If you just Google Duke POMMS, you'll generally find it. And there's a lot of material there about study design, the people on our team, and instructions for how to get in touch with us if you're interested in accessing samples or data. This project was really made possible by our clinical partner, Sarah Armstrong. So Sarah is the director of the Duke Healthy Lifestyles Clinic. And as you can read on the slide, this is a family-based weight management program for children and teens whose BMI is at or above the 95th percentile based on the age. And this is based in Durham, North Carolina. And the diversity of our patients in the study represent the diversity in Durham, North Carolina. So it's a very diverse group. And to date, the Healthy Lifestyles Clinic has served over 5,000 families. So, and Sarah's been very, outgoing with us and others and making, helping to initiate and facilitate studies in that clinic. The program does show good outcomes generally. Kids with at least 1 year of treatment show significant drops in BMI and body fat and blood pressure and cholesterol, as well as increased self-esteem and knowledge about nutrition. And there's an important kind of a wellness program component. Bull City Fit is a partner to the clinic, and there's also partnering with the Durham Parks and Rec for weekend activity sessions. So this is really the family-based weight management program. And so then collaborating with Sarah to work with her families, we initiated the POMMS study, and Jess is going to tell us a little bit about how the study was designed.
Yeah, again, thanks everyone. I'm Jessica. I'm going to go through some of the details of the study. And so the study was based on, excuse me, Dr. Sarah Armstrong's known patient population, and not necessarily designed for power to determine which treatments were most effective, but mostly which treatments her families tended to choose for their weight management and healthy lifestyles treatment. So recruitment included recruitment of healthy weight control adolescents. The criteria were they were at least age 10, BMI between the 5th and 85th percentile, and they had at least entered puberty. So this is also referred to as Tanner stage 2. And then adolescents with obesity were tracked with healthy lifestyles. And so all of Sarah's patient population have access to healthy lifestyles, which is family-based diet, exercise, therapy, and also social and emotional wellness. But some of the kids were also given options for medications. These include phentermine, Topiramax, which is an anti-seizure drug that has off-label weight loss indications, and metformin if the adolescents were showing early signs of metabolic syndrome. Some kids were offered a low carb diet and went on that track, and we did have individuals. So surgery is now offered to teenagers, and so some kids did have gastric bypass surgery. And so when we look at our recruitment numbers of who actually ended up in the study, we enrolled 71 kids in the healthy weight control arm. Adolescents with obesity were tracked into, the majority were healthy lifestyles, although in aggregate we also had 36 that tracked into the medication treatment group, 14 into the low carb group, and then 6 of them ultimately ended up having surgery. And so for the sampling process, what we ended up doing was that for the healthy weight control group, each adolescent had a single time point sample. And so these patients were recruited, or participants were recruited during their just general healthy wellness visits with their pediatricians. And so when they were recruited, they gave a single blood sample, fecal sample, and then had clinical measures, waist circumference, height, weight, blood pressure, those types of measures. And so we have all of these data on, for the majority, about 71 adolescents that were enrolled. For the blood samples too, some of them were stored. Actually, I'll tell you a little bit more about detail of that later. And then for healthy lifestyles, for the patients with obesity, they went through a 6-month treatment program. And I think we just, oh, there we go. down. Where basically at baseline, 3 months and 6 months into the treatment program, we took all clinical measures, a blood sample, stool sample, and then every 6 weeks throughout, they also had fecal samples taken. And so we have for about 140 of these individuals, a full longitudinal data set. And so when we look at the descriptive characteristics of our cohort, we were lucky that we finished recruiting right around the time of, finished the clinical studies sampling right around the time that COVID began and lockdown. And so we've been able to kind of undergo some of these analyses after that began. But basically what I wanted to tell you is that we're lucky that we have a very cool cohort, a few just hits on a few little details on our cohort. It does skew female for the treatment group. Females, according to Dr. Armstrong, tend to be more treatment seeking. It does skew the average age at entry is a little older for our healthy weight controls, although I don't think this ended up not being significant for our group. Adolescents with obesity do tend to enter puberty a little younger than adolescents within a typical weight range. And then the other important point, I think, is that we do have a highly diverse group, which we're very proud of. So we can really take a look at different, ethnic and racial factors involved in some of these outcomes. And so one definition that I'm going to use, you may be familiar with, BMI, just kind of using height and weight to calculate your BMI score as a measure of health. In adolescents is a little different, especially adolescents over in this 95th percentile and over. And so we use a P95 BMI to better characterize that group. And that's just defined as the BMI of the 95th percentile for age and weight. It's, sorry, the percentile of the BMI of the 95th percentile, right? So a child with a BMI at the 95th percentile would have a P95 BMI of 100. And so when you look at these numbers, that's why, that's what this means. And so adolescents tended to be around the 75th percentile of P95, and our adolescents with obesity were much higher. And so I understand that this audience is interested in the databases that are available, both for biorepository samples and data. And so just to give a quick rundown of what we have collected that will be available as part of our study. So we do have baseline and 6 months data for 140 adolescents with obesity, plus baseline data for 50 healthy weight controls. And that encompasses conventional metabolomics, I'm going to talk a little bit more about, targeted metabolomics, fecal rRNA gene sequencing on all of our available samples. That's over 700 or 780 samples. So anyone that gave us a stool sample that then did not withdraw themselves from the study at any point, we sequenced it all. So we've got 16S rRNA data, gene data on quite a bit. And then we also did shotgun DNA sequencing on our matched sample set for the adolescents. So if we had a full data set for baseline healthy weight in adolescents, then we also did their shotgun sequencing. So we have full, as close approximation of full genomes so that we can get a little more microbiome pathway and species analysis done on these adolescents. And then we also looked at fecal short chain fatty acids in as much of these matched sets as we had enough stool to do so. In our biorepository in total, that's public facing, we have over 3,000 serum and over 2,000 fecal samples that are can be shared. And there are also over 1,000 PBMC samples that are preserved in either DMSO or RNA later for any transcriptomics if anyone's interested. And then we do have an access request procedure in line, and that's available online, and a flyer advertising the POMMS biorepository. And again, I'm going to show our website a bunch of times and then again at the end, but if you just Google Duke and POMMS, you'll find it. And so just to give a quick brief outline of some of the clinical results. And so, our Dr. Armstrong, was telling us not to expect a lot of weight loss over a 6-month period, but we did see about half of the participants maintained or reduced their P95 BMI over the treatment period, although fewer saw reduction in another clinical measure of health, their HOMA-IR score. And HOMA-IR stands for the homeostatic model of insulin resistance, and it uses both fasting glucose and insulin levels to complete the score. And a lower score is more indicative of health. And so what I wanted to point out is that, we did expect our adolescents with obesity that received surgery to be amongst the group that did lose the most BMI over the 6-month period, but we also had a couple of kids that did well on just the lifestyle only. So diet and exercise management. And so when we look at a cross relationship of the percent change in HOMA-IR and the HOMA-IR score, and then also the delta BMI from baseline to 6 months, we see that the ones that had the biggest negative shift in both actually were the diet and exercise kids. There's no, just to kind of give you an idea, people always ask, who these kids are. And even though, this isn't enough power to really significantly say that this lifestyle treatment is effective, it did happen to work for, these individuals. And so one of the other things that we measured at baseline were things that you may be familiar with if you've been for your checkup, things like alanine aminotransferase, which is a measure of liver function, HbA1c or glycated hemoglobin. It's associated with insulin resistance, cholesterol, triglycerides, and then CRP, which is C-reactive protein. The interesting thing is that when we look at adolescents and children with obesity specifically, when they do, there's meta-analysis show that when they do lose weight, these measures of CRP and ALT do tend to drop. But at baseline, so when we just looked at our entry participants, adolescents with obesity had significantly, higher or lower values that trended, and this was similar to what we see in adults with obesity. The only difference we did not see was in just fasting glucose. And so what we are planning to now do ahead, we're just getting started looking at our longitudinal data at 0 and 6 months. Remember, these are just baseline data for the most part. It's what I'm going to show you. But our analysis include looking at any changes in these clinical measures of health with percent change in BMI, a percent change in HOMA, and then looking at these associations with both microbial taxa and the functional pathways expressed by those gut microbiota or present in those gut microbiota, and then also metabolomic measures associated with either adolescents with obesity or their healthy weight control counterparts. Okay.
Sure. Okay. So with that as a backdrop then on overall study design, as you can imagine, there are a lot of questions that can be asked within this data, and we've been wading into that. We're going to give you some preliminary results from some of those analyses. First, how does the gut microbiome and the serum metabolome differ between adolescents with obesity or healthy weight controls at baseline, right? Which is what Jess just talked about a little bit. And then also we can do the longitudinal comparisons, what features of the microbiome and metabolome correlate with weight loss intervention efficacy in the adolescents with obesity. And then in the end, we'll show you how we're also asking the question of whether or not those differences that we might see in gut microbiome might be sufficient to confer metabolic phenotypes to germ-free recipient mice. And again, this is all on a backdrop of a much larger body of knowledge about the answers to these questions in adults with obesity. And so again, what are the differences that we see between adolescents and adults with obesity? So to get started on that, we're going to look into the gut microbiome data. As just described to you, we generated 16S rRNA gene sequencing data, which gives you insight into the taxonomic lineages of bacteria that are present in different abundances in these populations or in these samples. And there's a number of different ways that these sequences can be analyzed. Without really showing you the data, what we were sort of surprised to see is that when we compared baseline, again, this is healthy weight control compared to adolescents with obesity at baseline, We did not detect any significant differences in several measures of alpha or beta diversity, which means the overall diversity of different bacterial lineages and their relative abundances to one another as an overall community were not significantly different between these two states. So, however, if we in more closely and ask whether there are specific bacterial types or ASVs that are enriched in one condition or another, we do see some bacterial ASVs that are significant. And here, they're kind of categorized by bacterial family, with with each color here representing a bacterial genus, and then each dot represents a separate ASV or separate sequence type. And so with these ASVs, we typically we're able to resolve genus level assignments, but rarely are we able to determine species level assignments. And so we don't know the precise identity of the bacteria, but we know that there are some bacteria that are enriched here, either higher in obese individuals or higher in healthy weight. And this is from 16S data. We are sort of in the early stages of analyzing the fecal shotgun DNA sequencing that just described to you. But one of the things that you can get from from the shotgun sequencing is a similar perspective on bacterial taxonomy and how different lineages are differentially enriched in one condition or the other. And so this is a similar plot, but using fecal shotgun DNA. And again, comparisons at baseline between healthy weight controls and adolescents with obesity. You do see some bacterial lineages that are enriched in one or the other. And the two highlights here, this Catenibacterium and here at this Intestinibacter, represent bacterial genera and perhaps species, or perhaps even ASVs that were detected in both 16S and fecal shotgun analysis. And so, this is an example of the kind of differences that we're seeing when we compare these bacterial communities at baseline. And so this is the Catenibacterium ASV, just to show you what that kind of data looks like. So this is using this 16S data, looking at this particular Catenibacterium ASV and looking at the relative abundance within each of these samples along the y-axis, and then individual samples are listed along the x-axis. And you can see that in healthy weight controls, this particular ASV was relatively low in the vast majority of samples, but in the adolescents with obesity, there's a higher frequency of appreciable numbers for this ASV. So again, there are others like this, but it does seem that the impact of obesity on the adolescent gut microbiome is really affecting a subset of bacterial lineages and not dramatically shaping overall community architecture. When we then begin to look at some of these longitudinal comparisons, we also see some differences. So here, this is comparing, looking at 16S data, either looking at their relationship to change in P95 BMI or to change in HOMA-IR. And again, we see some ASVs that are enriched in one condition or another. So it seems like it's quite possible that success or failure of the weight loss intervention may be correlating with certain bacterial lineages. This is something that we plan to explore more in the future. And so that's sort of the snapshot of where we are currently looking at the microbiome. And now we're going to switch to think about some of the metabolomic results.
I think there's a question in the chat. Does the recruitment data is before or during pandemic for the 6 months period?
The recruitment was completed before the pandemic. And I believe we actually did have our last sample collection about a month before lockdown.
Right, it's like January 2020 was the last sample. So we were, and that was just by luck, not by planning.
Yeah. So next I'm going to talk to you about we did collect a lot of metabolomics, and we've done some analysis on some of them, but I'm going to mostly focus on one story that we've think is kind of interesting and neat, and it has implications for obesity from, maybe a younger age up into adulthood. So one of the things that I want to talk about are, we were really focused on trying to identify metabolites of interest that were associated specifically with adolescents with obesity, and that could potentially predict treatment success or failure or lack of metabolic changes with the healthy weight treatment. And so one of the things that we know has already been associated with obesity in adults is circulating levels of the branched chain amino acids. And these are valine, leucine, and isoleucine. And so since the late 60s, we've known that these levels of these amino acids in circulation are associated with obesity, insulin resistance, and development of type 2 diabetes. And from the Framingham Heart Study, which monitored groups of adults for many years, and now is looking at their children and their descendants, they found that the levels of branched chain amino acids were actually prognostic for development of insulin resistance and type 2 diabetes as many as 20 years prior to incidence of disease. And so, and the other thing that's been shown is that these levels of branched chain amino acids in circulation go down after effective treatments for obesity and type 2 diabetes, especially things like gastric bypass surgery. And that there's other data that suggests that people with type 2 diabetes are likely to have bacteria in their gut that are especially able to synthesize branched chain amino acids. And we also know that you can give germ-free mice obesity-associated bacteria, and you will see an increased level of branched chain amino acids in circulation after that fecal material transfer into those germ-free mice. And so another related metabolite that we want to talk about is the branched chain keto acids. These are direct metabolites of these branched chain amino acids. And we also see that adults with obesity lower these levels following an exercise intervention or bariatric surgery. But we have much less data on the branched chain keto acids than we do on branched chain amino acids. Some of that is starting to emerge now in animal models of obesity, but in children and adults, we really don't have as much data as we do for branched chain amino acids. And so one of the questions that we had was, are there significant differences in these amino acids and their related metabolites in the adolescent population we're looking at? And so just a quick overview of what these are and how they're metabolized. So the branched chain amino acids are essential. You can't make them, you have to get them from diet or potentially from your gut microbes. And they're made by bacteria and by plants. And so you can see that they are leucine, isoleucine, and valine, and they are first metabolized by this branched chain aminotransferase. This is a reversible reaction so that you can take these amino acids and turn them into the keto acids, or they can be reversed. This process can be reversed, and these these amino acids can then be used for things like muscle building, protein building, those kinds of things. This process requires glutamate to take, it requires alpha ketoglutarate, and the processing of glutamate, if there is excess ammonia, then glutamate becomes glutamine to detoxify that ammonia. And then the other quick thing I wanted to tell you is that once these keto acids are dehydrogenated by the branched chain keto acid dehydrogenase, this is now a one-way street. These molecules are now trapped in the mitochondria. They become food for the TCA cycle to generate energy. And so if we look at this enzyme activity, we can really try to figure out, how are these amino acids being treated, and then how are they being metabolized? And so this branched chain keto acid dehydrogenase, I'm not going to talk about it today, but we actually know a fair amount about how it's regulated. And it's interesting, so it's regulated differently in the tissues, in liver versus fat tissue versus muscle tissue. And so this is a kind of a cool story that we can really try to follow up on in these adolescents. And so for right now, though, I'm going to focus on the keto acids. So these are keto isocaproate, which is the keto acid of leucine, keto methylvalerate, the keto acid of isoleucine, and keto isovalerate, which is the keto acid of valine. And so we published an interim paper looking at a very small subset of our participants as they were being enrolled, 27 healthy weight and 27 adolescents with obesity. And we saw this nominally significant, but not after adjusting, difference where the kids with obesity had higher levels of valine, but weirdly lower levels of these keto acids. And so in adults, what we would expect is if there's high levels of branched chain amino acids in obesity, they also have high levels of these branched chain keto acids. But again, the studies are limited. And so what we wanted to do was see if we could, if that would capitulate once we analyzed our larger study. And so what we did was we looked at all of our targeted metabolomes, metabolomics. This is over 245 baseline samples are some of the data I'm going to show you today, 195 adolescents with obesity and 50 healthy weight control. I'm going to pretty much focus on the amino acids and, branched chain amino acids, but we did look at over 65 metabolites, combined panel of amino acids and acyl carnitines, the keto acids, and then related 3-hydroxyisobutyrate and 2-hydroxyisobutyrate, which are products of valine catabolism. And so when we look at these data, what we saw was that there are, when we look at this plot, this is the fold change of these metabolites between adolescents with healthy weight control versus adolescents, the healthy weight control adolescents and adolescents with obesity. And so the numbers above this line are the things that had a significant p-value being associated with either higher in adolescents with obesity, or if they're shifted this way, they're higher in adolescents with healthy weight control. And what we saw here again was that valine was shifting this way, but here are the keto acids, which are going in the opposite direction. And then when we look at serum levels of these amino acids with association with the insulin resistance score at baseline, what we saw again was that valine and now leucine and isoleucine are also significantly associated with this signature of having a higher insulin resistance. And so this is consistent with what we see in adults, but we really wanted to dig into what these keto acids were doing. And what we found as we essentially replicated what we saw in our interim analysis, where in these graphs, I'm showing you the aggregate data where adolescents with obesity had a significantly higher level of circulating valine than the adolescents that we recruited for healthy weight controls. But that the keto acids, interestingly, my little cheat sheet here, of isoleucine and leucine were lower in adolescents with obesity. And so trying to figure out what that meant. And so when we looked at the leucine, isoleucine values at baseline, they were actually not significantly different. One thing about our metabolic mass spec panel is that these are so closely related in mass and charge, they actually measure as one. And then when we looked at keto isovalerate, we saw again, there was no significant difference. Remember, this is the keto acid of valine. And so what we wanted to figure out is what is driving the signature. And we had gotten a hint that some other people were looking at similar data sets and we're seeing pretty strong sex differences between some of these metabolites. And so when we looked a little more closely, we did see that there are sex differences for related branched chain amino acids. Even in the absence of baseline differences between cohorts, we were seeing differences in sex. And so the way these are color coded is that the males are triangles, females are circles, and the healthy weight control data is in orange. And so when we look at all males versus all females, we see again that the valine is actually higher and that in healthy weight control, often the signatures were driven again by males having higher levels here. But we didn't see any significant differences between males and females when we just compared them amongst the obese cohort. When we looked at leucine and isoleucine, we saw a similar story where males higher than females taken in aggregate. No significant differences in the healthy weight control, but then the males with obesity were higher than females with obesity. And then we see the male healthy weight control is again higher than the males with obesity here. And so when trying to kind of parse this out a little bit, in general, we also saw the same signature in keto acids, and this really, again, seemed to be strongly driven by the male healthy weight control, which in both cases was much higher in the male group with healthy weight control, but although we did see more significant differences between these groups, with males in general having higher relative values. And so when we looked now, and if you remember, valine was one of the ones that was higher in adolescents with obesity versus healthy weight control at baseline, while its keto acids showed no difference at baseline. And when we looked at the keto acid of valine, we also saw that this had this signature where males were significantly higher, but we also saw males had higher here too, whereas there was no difference in the male healthy weight control. And so one of the things we're trying to figure out now is how are these processes decoupled potentially? And we think it's probably happening in the muscle. So a lot of biology to kind of talk through. But that might make sense for males undergoing puberty. They just have potentially more muscle mass and could potentially protect from overloading their mitochondria with too much energy at that point in the muscle. And so our hypothesis is that the increase in branched chain keto acids in adult obesity is related to that. Remember, I told you that the branched chain keto acid dehydrogenase is what takes those branched chain keto acids and turns them into energy for the rest of the TCA cycle and other things that they can contribute to ATP generation in the mitochondria. But in obesity, we know that this enzyme is actually inactivated. And so the muscle tends to have to take over with excess branched chain keto acids and either use them to build muscle or use them for energy. And that, if for a long cascade, ends up with excess glucose. And you can see if you have a lot of energy overload in the mitochondria, you may end up with excess glucose and leading to the beginnings of insulin resistance. And so the other things that we wanted to take a look at that feed into this pathway are glutamate and glycine. And what we hypothesized that would fit into our hypothesis for adolescents with obesity to kind of keep this BCAA level in check was to look at an increase, look at the glutamate levels, and an increase in muscle glutamate could potentially drive reanimation of these keto acids to branched chain amino acids and cause a dissociation of this metabolism. But we should also see a depletion of glycine as it feeds into the ability of glutamate to be to help detoxify the excess ammonia and become glutamine. And so because again of our panel, we measured glutamate and glutamine as a single level, but we did see that we do have a higher level of glutamate in circulation in adolescents with obesity, as well as a depletion of glycine. And so that does track with again, this is just a hypothesis. So we're trying to come up with ways to test this in animal models of obesity in adolescents. And so I should also say that when we analyze the differences in these other amino acids, it does seem to be that case versus control status, or the obese status does seem to be driving these differences, as there's no obvious sex differences in these. And so we're still thinking about, well, what could potentially be driving these other muscle or other potential amino acid changes and this dissociation between males and females in some of these signatures. And so one of the things that we do have access to is a data set that was run at the same time as ours that has younger kids. And so we wanted to see, is this higher branched chain keto acid signature in age or a puberty dependent phenotype? And so in an associated data set, we have access to all of our samples were actually run at the same time. So they were able to leverage the infrastructure that we created with the POMMS study. And we used pooled metabolomic and rRNA gene sequencing analyses between the study that looked at younger kids. And so we can actually use it as a validation set to look for sex and age dependent factors here. And so if you look at the age of this recruitment set, this was focused on cardiometabolic factors. So these kids all had an exercise intervention and more fitness trackers. It's really cool. They had some cool outcomes, and this ran during COVID. So if they had some really neat data changes around COVID that are unfortunately kind of sad, but save that for another time. So if we look at the age group here, they skew much younger than ours, right? So most of the kids in this study were between 5 and 11, so had not yet entered puberty. And so when we look at keto acids by sex of the entire cohort, remember it goes to 18, we do start to see signatures between these branched chain keto acids where males in general are higher, and then females for both the keto methylvalerate and the keto isocaproate. So this tracks with what we saw in our cohort as well. Keto isovalerate, not quite there yet. But when we look at just a younger age group, which we can posit has not entered puberty, we don't have puberty status data on all of these kids, but we can impute that 5, 6, 7, 8-year-olds are not likely to have entered puberty. We can see that there are no significant differences between these data, so they have not yet emerged potentially. And then if we look at aggregate, there's no significant difference here. And so we are in the process of analyzing by age and sex here. But you can see for each individual age group, we are not highly powered to make any strong computational decisions. But if we combine our data sets, then we should be able to make some interesting associations here. And so the last step. Yeah.
We can do it together.
Okay.
So the last part, the last piece of data that we wanted to share with you today is sort of the status of our efforts to look at the sufficiency of the microbiomes associated with adolescents with obesity to confer those phenotypes to mice. And again, hopefully we're all aware with the significant number of studies by several different groups that have shown that obese phenotypes can be conferred from obese donors to germ-free mice by fecal transplant. And this has been seen coming from human donors in several studies, and it's even more consistently observed in mouse studies where you bring in mouse stool from obese donors. But a really important thing to recognize is that practically all of those studies used adult donors, where the state of obesity had been established for, a very long time, and presumably the microbiome was sort of highly adapted to that obese state. And so what do we see in adolescents where that state has been around for, a short amount of time? So does fecal material from adolescents with obesity confer obese metabolic phenotypes to germ-free mice? So we have been doing a set of fecal transplant studies using C57 black 6 male germ-free mice with 4 or 5 mice per FMT. And the study design is shown here with fecal transfer at day 0 and transfer into an isocage at day 0, gavage in the fecal material, and then weigh them for several days out to a 14-day endpoint, where after a short fast, we harvest and weigh them, collecting a variety of tissues and samples for downstream analyses. And so far, we've been testing individual donors from the POMMS study, and we're up to 17 so far. And remember, when many groups have done this using donors from obese adult humans or mice, they do see an increase in body weight and fat pad weight, at least temporarily after the transfer. But that's actually not what we've been seeing. So here, the healthy weight control samples are in orange, and the OB samples are in black. And you can see with percent weight change from day 0 along the y-axis here, that you see a range of responses, and but overall, those average out to be not significantly different between the 2 different donor groups. And we see that whether we look at mean body weight gain over time, or the mean final weight of epidermal fat pads. And so, so again, even though there may be some individual donors that are able to evoke that effect, and this is something that we're considering exploring more, overall, it does look as though the adolescent gut microbiome may not be able to promote adiposity in recipient germ-free mice, which is distinct from what we've seen, or from what others have seen in using adult microbiotas. So with that, we're going to summarize briefly. Here are some of the things that we talked about. As we've seen, adults with obesity do have a surprising and seemingly unique BCAA-related metabolomic signature compared to adults with obesity. We think this is really interesting. It underscores that adolescents are not simply little adults, that there's different physiology happening there, and that in that transitional state, we may be able to learn something about these the origins of adult obesity. And this is something that we hope to explore more in the future, both in humans and in animal models. We've seen that some of the amino acids and keto acids have significant sex and possibly also age differences in adolescents. And this is really forcing us to really look at all these different data types as a function of sex and possibly also age, which makes it slightly more complicated. But we're hoping that with being aware of those potential differences, it's going to help us conduct a better analysis. We showed you how even though the overall impact of obesity on the microbiome doesn't have a dramatic effect on the overall community structure, that there were certain bacterial taxa that were differential at baseline, and also with change in BMI and HOMA. And so we're very much in progress there, and we're hoping to look more closely in the weeks and months to come at microbial species and strains and functional pathway associations using our shotgun data to see if we can identify correlates with other markers of health and metabolic signatures. And then again, we're sort of in the midst of our FMT studies, but it seems that the early result is that the ability of the obesity-associated microbiome to promote adiposity may not be attained until adulthood. And this is interesting from a perspective of host microbiome interactions. One possible interpretation there is that over a lifetime, that the microbiome becomes more attuned to the physiologic state of the host and begins sort of causally contributing to the metabolic state of the host. But perhaps during adolescence, that adaptation hasn't fully occurred yet. And so as we see these sort of adaptive metabolic profiles in the host, maybe we're also seeing kind of an adaptive state also in the microbiome as well. And certainly we're looking forward to doing more longitudinal analyses using all of our data types in hopes that we might be able to come up with some new predictive indicators of who will respond to therapeutic intervention or not. And so stay tuned for more, hopefully, in the months to come. And so with that, I think we'll stop and open the floor for questions, and just remind you that if you have more questions about or curious about accessing the samples or data, please feel free to get in touch with us at this link. We'll stop there.
Okay, thank you for the presentation. So there are several questions in the chat now. The first question is from Dr. Chen. Thanks for the beautiful data and presentation. I have two rather naive questions. First one is, are the major differences in terms of microbiome and the BCAA changes between different interventions, in particular lifestyle vs medication?
Yeah, we unfortunately we're not powered to answer that. We tried by potentially aggregating our kids, adolescents that were on the drug treatment to look at their fecal samples. We've only really looked at baseline though, so at that point nobody had started the drugs yet. But so far, just looking by eye and by doing a couple brief analyses, we're not really seeing a big effect of medication yet. And so I will say, too, of those 36 kids, there's probably 4 different medications there. And we know that metformin does affect the microbiome pretty significantly, at least in adults that it's been looked at. But we're not actually seeing that. And it could just be that the kids, it's not enough time potentially for them to have been on it for it to have been major change. Also, we don't have any measure of compliance really for any of the treatments. So we don't know, how well they were taking their medications, all those things. We don't have those measures. And so we're still looking at those data, but and so far I don't know that we'll have enough power to see how much medication had an effect on microbiome. But we've really only looked at baseline, and at that point, there are no medications had been started at that point.
Yeah, and Jess may have mentioned this, but the numbers that we were able to enroll into those different groups were not, it was based on the historic enrollment patterns in the clinic. So we really weren't able to, for example, enrich or try to match the number of participants that were in any one drug group or in the low carb diet, for example, compared to the healthy lifestyle. So we were sort of at the mercy of who was enrolling in the program. So that kind of, that limited our numbers. The second question is, you present the changes mostly relative to HOMA-IR. Do they also correlate with other changes in body weight, or do the latter two correlate well?
Yeah, so the body weight changes, we were actually kind of shocked. We weren't, the clinician had told us not to expect a lot of body weight changes, and her goal is actually more metabolic health change. But I think because the body weight changes, we basically just took everyone that had any change that was anything below 0, so it may not have been significant enough to actually affect insulin resistance. Whereas for metabolic health, that's one of the major things that she can measure is change in insulin resistance. And so if you look at the rightmost plot of the percent change in HOMA versus the percent or the change in BMI, you can see there really isn't a super strong correlation except for, those ones that had both negative changes. So there weren't that many individuals that saw strong decreases in both.
Yeah, and then this bit of data here sort of addresses that question a little bit too. You can see how the change in P95 BMI relates to percent change in HOMA-IR. So there is a relationship there.
Okay, thank you. There is another question from Hiba.
Yeah.
Why did you choose male mice instead of female for FMT? I wonder if you are not seeing a change because of that HOMA-IR usually correlated well with percentage of body fat as measured by DEXA scans.
The second part was in relation to that first question by Dr. Chen. So yeah.
Okay. Yeah, it's a good question. Obviously, our data that we showed you today are amongst many that shows important sex differences in some of these metabolic interactions. And so, I think there are a couple of reasons. One is that the accessibility of epidermal fat was an important part of our endpoint. And we wanted to be able to access that readily. Also, these FMT studies, as anyone who's done germ-free mouse studies, that they're expensive and cumbersome, and to try to match numbers of males and females evenly across the treatment groups was just more than we could manage at the time. So we had to pick one or the other, and we picked males. But it's a fair point, and I think the farther we get into this, especially as we continue to wrestle with some of these sex differences we're seeing in the keto acid branching amino acid phenotypes, I think we're going to be forced to go back and do at least some of those FMTs in both sexes and see if we see anything different. So it's a fair point.
Yeah, and as far as the percent body fat for the kids, we don't have those data. We did try, I think for most of them, we have waist circumference as another, good correlative measure, but we don't have percent body fat scans for those kids.
The next question is a technical question from
Sure.
the audience. How do you do the fecal microbiota transplantation? How many times do you?
Sure. Do you want to?
Yeah. So we make a slurry that's 10% fecal material and 90% reduced PBS. And then they get a single gavage of that slurry one on the day of transfer. And so we've piloted a few different ways to do this, mostly to try to keep contamination down while still, reducing the amount of stress that the animals are undergoing. So yeah, so we just do a single gavage. And so what we have not yet looked at is the retention of the microbes that at the end of those 2 weeks. So we've got samples to analyze what went in versus what came out the 2 weeks and which ones actually engrafted. Yeah.
Okay, next question is from Corinne. Regarding longitudinal data, do you have the opportunity to follow these adolescents cohort?
Yeah, sure. It's a good question. I think we've asked the same question of our of Dr. Armstrong, our clinical partner, and it's not an impossibility, but it's not a part of the IRB and study design currently. So they would have to be re-recruited, re-consented as a part of a new study. But it's if someone was interested in that sufficiently, we could certainly have that conversation and see what the feasibility was. Ours is as we introduced, the Healthy Lifestyles Clinic has served a lot of families, and so even beyond our particular study and the data that we're generating, I think Dr. Armstrong's kind of collaborative network have generated a number of different types of data from different sub groups coming out of the Healthy Lifestyles Clinic over a few years. And so I think one could ask this question about longitudinal or kind of follow-up recruitment could be done for this cohort, perhaps for others, like the AHA cohort that we talked about, which is kind of conducted sort of separately, but at the same time as ours, that could also be included in that kind of follow-up. So we have not tried, but it sounded like it could be tough, but if someone was interested, we could certainly look into it further. Does that sound about right?
Yeah, definitely. And for the last question on the short chain fatty acids, so we do have serum to potentially look at incretin hormone associations with short chain fatty acids. I will say that we did not see a lot of differences in short chain fatty acids between the cohorts, but that's definitely a possibility, and we're aware of those connections in the literature. So yeah, that's a great question.